This video is on question 47 from the 2012 IMAT paper. In this question, we're told that gene expression can be regulated by one of the following: transcription factors, DNA replication factors, RNA polymerase, rough endoplasmic reticulum, and the position of the genes on the alleles. So starting with transcription factors, so what exactly are transcription factors? Well, they're proteins which bind to the promoter region of a gene. And this binding of transcription factors to the promoter region allows RNA polymerase to bind to the promoter region to initiate transcription, to enable the mRNA strand to be synthesized. Therefore, yes, transcription factors can regulate gene expression, because if transcription factors don't bind, RNA polymerase will not be able to bind to the promoter, and so the gene will not be transcribed, and therefore won't be expressed. So now, even though we know transcription factors are our answer, I'll still address the others for completeness. So, DNA replication factors. So this might not be something you actually cover at A-level, but you can deduce from the name that these are involved in some way in DNA replication. And in reality, they're involved in controlling DNA replication and determining how many times the DNA will replicate in one cell cycle, and so often DNA replication factors will ensure that the DNA only replicates once in one cell cycle. So no, it's not DNA replication factors, as DNA replication doesn't really have anything to do with gene expression. Next we have RNA polymerase, and what is the role of RNA polymerase? It carries out transcription, and so that is producing an mRNA strand from the DNA. And so while this does control the production of mRNA, which is then translated into a polypeptide, which then multiple polypeptides assemble as a protein to allow the gene to be expressed, RNA polymerase depends on these transcription factors binding to the promoter first. So RNA polymerase just does its job of transcription, and it's really the transcription factors which regulate the gene expression. Next we have the rough endoplasmic reticulum. Now, the rough endoplasmic reticulum is an organelle which houses many of the ribosomes in eukaryotic cells, and it's these ribosomes which carry out translation, which is the conversion of an mRNA strand into a polypeptide. And so, while its overall function is for the synthesis of proteins with the ribosomes and the modification of proteins, the rough endoplasmic reticulum itself does not really regulate gene expression, as the gene expression is generally regulated further upstream, meaning before in the process with these transcription factors. And for our final point, we have the position of the genes on the alleles. Now, this point doesn't really make sense, as alleles are a form of a particular gene, and so you can't have genes on alleles. This doesn't really make sense. So no, it's not this option either. Therefore, our answer is A, transcription factors.